Once again, good morning, everyone. Once again, I welcome you to this special lecture on ecological and biodiversity sampling that I have arranged actually again for my students from the UPV Division of Biological Sciences, which is hosted by me, the instructor of the ecology laboratory course now this term. So before I introduce our lecturer for today, let me review some of the house rules that we will have in this lecture session. So of course, first, we need to follow basic etiquette in communicating with the guest and the host. Hopefully, everybody keeps their cameras off and their microphones on mute while the lecture is ongoing. Once again, once Dr. Ohara starts his lecture, we will no longer admit other participants to the session. So if the other people we still want to watch this lecture series or lecture, it will be available via YouTube after this session has culminated. Questions and comments can be posted in the chat box when those signals in the chat box that you may do so. So we will have a question and answer segment on this lecture. Do indicate your name and the institution that you belong to when you give your questions and comments in the chat box. Sorry. And of course, once again, the session will be recorded and I can give you the Google Drive link if you wish to download it, but the session will also be posted in YouTube if you want to watch it online. So much for that. So once again, I would like to introduce the one that I bothered for this lecture. No, I thank and apologize to Dr. Ohara for this lecture. Again, for my students, it seems that I'm a very lazy instructor because I ask other people to lecture on my class. But anyway, so Dr. Masahiro Ohara is the Vice Director of the Hokkaido University Museum and also the Collection Manager for Systematic Entomology of the Hokkaido University Museum. Dr. Ohara is also a professor in the same university for systematic entomology and as well as for agriculture in Hokkaido University. He is also the editor-in-chief of the European Languages Journal of the Coleopterological Society of Japan. He took his PhD in the Hokkaido University Graduate School. He's also an associate editor for entomology for the Philippine Journal of Systematic Biology, which is I am a managing editor of. That's the reason why I know Dr. Ohara. He had at least published 60 scientific papers, plus 27 popular and semi-popular articles, and 3 books on the systematics, functional morphology, distribution, zoogeography, and behavior of insects with emphasis on coleopterans of the family Hysteridae. So, Dr. Ohara is indeed a world-renowned expert when it comes to sampling beetles. So, without further ado, I give you Dr. Masahiro Ohara.
Okay, thank you very much. So nice introduction. I want to start to my PowerPoint. May I share it?
Yeah, you may start sharing your screen, Dr. Ohara.
Okay, can you see my slide?
Yes, Professor, we can already see.
So good morning, everybody. My name is Masahiro Ohara for the Ericsson. Thank you very much for the nice introduction. I'm also very happy to be here for this so nice invitation. I'm very happy for such opportunity to talk about for the insect sampling method. Yeah. Okay. So today's my topic is 3 topics. So first is Entomofauna, the monitoring survey. Oh, it's not so easy to move. Yes, and the Beetle taxonomy and the museum collection. Okay, so first of all, it is better to my introduction and the nature and the insect of Hokkaido, Japan. And next, I want to talk the 3 topics. So this is my CV, and I was born in Tokyo. It's 1961, and I graduated to Kagoshima University. So because it's my supervisor is Takehiko Nakane, he's a very famous person of the Coleoptera. And after for the undergraduate, I moved to the Hokkaido University and master course and doctor course. It's my supervisor is Sadao Takagi. He's a specialist of the scaleback. I got a PhD in Hokkaido University. And so during the doctor course, I received for the JSPS fellow. So after graduate school, I got a job to the Otaru Museum as a curator, 6 years. And after 1997, I moved to the Hokkaido University for the Faculty of Agriculture as an assistant professor. And from 2000, I moved again to the Hokkaido University Museum as an associate professor. Since 2011, I have been for the professor in the museums. So last 2 years, I'm very busy for the administration work because it's a society. It's one of the Coleopterological Society of Japan, and the other is the Entomological Society of Japan. I'm now president, and so much for the work of the administration. So this is my university, Hokkaido University. So about 145 or some for histories, and it's at Sapporo, Hokkaido, Japan. And the Sapporo city is very famous of the Winter Olympic city. It's 1972. So Sapporo is here, is Hokkaido. The Hokkaido is the northernmost island. And so this is a map of the Japan, but you know, for this is upside down. So here is a Hokkaido. And so you can see for the 3 straits. So one of the this is a Sakhalin Island and Hokkaido. It's not so deep. It's 60 meters. So during the glaciers time, this is 2 islands continued, and so many mammals and so animals, plants move from here to north to south. And the Tsugaru Strait is rather deep, 450, and there is a barrier between for the Honshu Island and the Hokkaido Island. And we have a different black bear and brown bear. So this is a very important biological, biogeographical barriers. And here is a Basalt Strait, is very deep. So this is also important for the marine because this is Okhotsk Sea and open to the, so even for the long time, for the open to for the Pacific. So of course, it's Hokkaido and Japan. This is one of the Holoarctic region and the Paraarctic region. And in the Hokkaido area, we think is basically it's a Manchurian subregion, but there is so many elements of Siberian subregion element for zoology. So this is our museum's exhibition panel. So there is so many biogeographical line, and in Hokkaido there is 4 line, and from the Sakhalin, the Schmidt line, the Hatta line, and the Miyabe line, and the Kono line, and the Chromatznai lowland, and the Blackiston line, and the red one is a person name, so who is related to the Hokkaido universities. So this is a Hatta line. It's related to the reptile. It's a different fauna of the reptile. So Miyabe line, this is a botanical line, is quite different so vegetation. And the Kono line is related to the butterfly, and the Chromatznai lowland this is a distribution, is the northernmost distribution of the tree of the fungus. So this is the Blackiston line, is very important for the animals. So this is a view of the Hokkaido. It's a Shiretoko is one of the World Heritage Site. So there is so many deer and bear and so many sea mammals. So this is a forest of the Erimo area. So one of the Carabide beetle, this is the Hokkaido endemic species. And we can try to do a light trap. So we got so many for the Lucanidae. It's a very big beetles. And this is a coastline. And so of course there is an artificial material for the protect for the wave. So we can collect is under seaweed pile. So some pile of the seaweed is the deep of the water, and is an insect flowing of the water, and we can pick up to the so many insect. Also sometimes it's shifting here. Yeah. So recently I conducted to for the coastline littoral beetles, and I collected so many places of the Hokkaido with my graduate student. So this is a Lilozoma, is a very northern element beetles, only distributed to the east part of the Hokkaido. So thus beetles is distributed also Kuril Island and Kamchatka and Aleutian. So this is a littoral hydrophilic beetles is Celsion. In Hokkaido, there is 6 species. So one of them is Celsion is a symbion, only distributed east and north. This is typical for the northern element of the insect. So this beetle is very curious. It's a genus is Aegialitis, it's a family is Salpingidae. So they live in such rocky shore, and there's a crack of the rock, and they living here. It's a very curious, and without hind wing, they cannot to fly. The genus is distributed from Hokkaido to Kuril, Aleutian, and Alaska, and West Coast America, and until California. This is a very good target for biogeography. So recently we work of the molecular and morphology of the genus. So this is my beetles with Hysterics. So this species also is only for the north and east part of the Hokkaido. This is one of the northern elements. Oops. So this is a butterfly, very beautiful butterfly. This is a national treasure, only distributed to the top of the mountain of the Hokkaido, Daisetsu Mountains. So there is another Carabide, very beautiful one. So this one is only distributed for the island of the Rishiri. And it's a very high mountains, and only the beetles are only distributed to the top of the mountain of the island. So very recently we found these beetles and a new subgenus. So last one is just a Diptera, is a Tipirinidae. This is a wingless fly, Diptera. So they walking for the snow. So we can find only winters. This is a male, and this is very curious, it's a Diptera. So just as I show for the some insect on the Hokkaido. So I start to do for the method for the collecting. So first topic, survey of Entomofauna. So 1987, I conducted the survey of the this forest is a Nopporo Forest Park. So 1987, that times I was for master course student. Every week I visit this forest and setting the trap. So in the Nopporo is here, is our university here, about 1 hour by car, I can arrive to the Nopporo Forest Park. I set this trap. This is for the bait trap. So gauge is inside set to the chicken meat. and the chicken meat is decaying, and the fly coming, and there is so many maggot. And my beetle is a Hysteridae, is a predator of the maggot. And I can collect so many for the Hysteridae beetles from the trap. So this is a result from April, from spring to the autumn. So each week I collect Hysteridae in the forest, and I can realize it. So 11 species, 11 Hysteridae beetles in the park, and each species is different over the seasonality. For example, is Margarinosus cadavericola, only we can find of in autumn. That is very interesting for the seasonality of the Hysteridae beetles. Yeah, also I can find one new species. This is Japanese name is Nopporohime emmamushi, and the Latin name is Margarinotus ezoensis. Ezo is old name of Hokkaido. So I was in first year of the doctor course, I described this new species. So I graduated for the graduate school. I got a job with the Otaru Museum. So this is Otaru City, and that is a local museum. And so local museum should be work of the local fauna in the city. And I started to the survey of Entomofauna of the city. So this is for the bulletin of the Otaru Museum. I reported several work of the local fauna. So it's a very small area of the Naibo area. It's such so nice forest. And each week I visit this forest and the setting for the several trap. So one of the pit hole trap, probably you know this is a very typical for the insect trap. Just is a cup on the ground, and we can collect it some carabide or walking beetles. So this is a light trap. So we can collect is moths and beetles and fly and many kind of the noctic for the insect. So this is a yellow pan trap. Very simple but very effective. So just this is a pan of with the yellow colors and water in here, and so many insects is coming here and hold in the waters. So this is a result of the yellow pan. And usually for the temperature zone, so everybody think is summer is a very good number of so many insects in the summer. But as a result, so it's November, there is a most highest number of the individual. So this is a diptera. So before the snowing, the Hokkaido forest is so many insects with very tiny midge is a fly is coming. So this is a very interesting result. Also, we collected general collecting, and also I did to the beta trap, and I collected so many history here. So this is an exhibition of the Otaru Museum. So last year, 2 years ago, so they making for the, this is a Naibo Park. And so this is a history of the park, and for the survey of the insect, also is they are research of the plant and the bird. So there is a exhibition of the how to collect insect for show for the general people. This is a pit hole trap and a yellow pan trap, and this is a diorama, small diorama of the light trap. So next, I also is another area, so Okusawa area, and I setting for the, this is a marriage trap. Probably you know for the marriage trap, this is like a tent. So it's a roof part is white, and many insects come here and fly, and the hole is in the bottles. Also, this is a very effective trap. So this is a list of the paper of the survey, and sorry for the Japanese, but this is Odonata, Plecoptera, Orthoptera, Hemiptera, Coleoptera, so number of the species, and for the, this is author of the papers. Anyway, for the in the Hotel Museum, so we conducted the survey and make so many small paper for the fantastic entomology inventories. So after Hotel Museum, I back to the staff as a staff of the university, and so one of my student graduated at the university, and she worked for the Obihiro museums, and she asked me to for the survey of the local fauna. So this is Obihiro city, and so two places, Oyama Ryokuchi. Yeah, Obihiro is here, of the center of the Hokkaido. Yeah, this is just a schedule for from 2008 and 2012. We are once or twice or three times sometimes. So we checking for the fauna of the Obihiro city. Yeah, this is Obihiro city, and a very small area, but very important for the forest for the city. And we is research of the fauna. Yeah, this is a view of the spring. Yeah, just after for the melt of the snowing is so many like a marsh, but in August is all dry and it's quite a different looks in the view. So this is May, so many flowering, but in the middle of summer is like a bush. So we collect, this is the general way they're looking, and we can collect is a rather big insect, is a butterfly or cicada. Yeah, it's a common butterfly in the forest. Also cicada. And the same place, also we use our insect net. So we call it the sweeping. And anyway, we sweeping for the grass and the tree of the leaf, and we can collect so many insects. So, but it's a fly and a wasp, easy to fly from the net. So we, so how to say, covered to the net and use for the, so this aspirators, and we picking up and so small insect. So this is one of the result of the sweeping, and you can see for the Neuroptera, Trachyptera, Plecoptera, Dermaptera, it's so many kind of insect we can collect. So this is 4 major orders. It's a Hymenoptera, Hemiptera, and Coleoptera, and Diptera. Usually Hymenoptera and Diptera is much common, but I'm a Coleopterist. I collected so many Coleoptera. So this is some specimen of the Coleoptera beetles from the forest. It's all photo is different families. So this is one of the example. Is we conducted survey in 2008, but before that there is a last survey. It's 1999. So that times for they collected 14 species of Chrysomelidae. So sorry for the Japanese names. And so of them is 4 species we can collect again in 2008. But we more, and we can collect these Chrysomelidae beetles, and the total is 21, and additional is 7 species. So I don't know for the result, why is the differences, but maybe for the habitat of the forest or park, vegetation is a change, or maybe for the research method is a little bit different. Anyway, so we can make an inventory with 21 species. So this is one of the monitoring. So 10 years ago, is a Chrysomelidae fauna more poor, but now is a more species. So this is one of the result of the monitoring. So we also check with the marsh area. So we can collect its aquatic beetles, also is Odonata, the Plecoptera, and such as small streams. So this is ant nest. Yes, we can collect ant, but not only ant, but also, yeah, I use for the shifting. Yeah, there is so many ants. And included for the ant nest, we can find is different creatures. So this is a Pseraphidae of the Myrmecophilus beetles, and this is a Staphylinidae. So this is a very interesting beetles. Also, they live in only ant nest, and each ant has a very specific for the such beetles. That is very interesting. Also, it's very so many new species. Yeah, we making again for the bait trap. So this is a member of the bait trap. So this is a Hydrophilidae and Hysterics and Staphylinidae and Sylphidae and Beetleidae. Also is a Scarabidae members. So next is Elimo town area. Also Elimo town curators ask me for the survey of the insect fauna. Yeah, Elimo is a southern tip of the Cape of the Elimo. Is a more tropical element here. Yeah, this is a course of the survey and the schedule. It's only 1 year, but we frequently visited the area. Yeah, it's a very nice place. So a small lake of the mountain. So this photo is not clear, but its shape is a heart. It's very famous as a lake. We set is a marriage trap and bait trap. So this is for a photo of the bait trap. This is the bait trap and the shaking of the gauge, and there is so many for dropped of the beetles. And I picking up to the tube. Yeah, this way. Yeah, and sometimes I collected the soil around here and making for the bell laser trap. This is a light trap. Yeah, recently we use LED. This is automatic, and overnight we collected so many insects inside here. Yeah, I checked to the rocky shore. Also is underbark. So underbark is also important for the Hysterics beetles and other beetles. Yeah, this is the last for the Hokkaido University campus in my university. Yeah, so this is a very urban, is for Sapporo city, is a huge, about 2.5 million people. And the Hokkaido University has a little bit forest, but it's a nice forest. And yeah, so just here, it's a very small wet area. It's like a small marsh. And so we checking every year here. So this is a ladybird beetles. The species is a red data book species. So in Sapporo still we have this beetle, but other prefecture in Honshu already extinct or endangered. Yes, this is endangered mapped, and some prefecture is very endangered. Also, this is a Carabidae and small hydrophilic beetles. Also is an endangered species. So many city people don't know for the beetles, but is entomologist checking for the every year. We can show it's a very small nature, but it's very important for the city and the people. Also in the campus, there is so many wasps. It's a very big wasp. It's a very danger. But so you can see here, this is one of the insect, very rare insect, female of the Strepsiptera. Yeah. So this is a parasite insect. So inside of the body of the wasp, this is a female insect. Yeah, it's such shape. It's very strange. It's all reduced for the leg and the wing or outside of the body. Only they keep to the hard part of the head. Sorry, Japanese. This is a Strepsiptera. This is a life cycle of the Strepsiptera. This is a female body. And you show, yeah, you can see for the photo is the last one. And also we can find for the male. So this is a parasite insect. It's very, very rare, but every year we checking for the forest. It's so many entomologists for the professor and graduate student collecting. Yeah, sometimes we can find for the very strange rare insect. So this is just for the schema. So we research for the local nature of the local area. So that's nature, it's just is natures. But we want to understand for the natures, how to understand for the local natures. The one way for the, we sampling specimen extraction and make for the specimen collection in the museums. And so we really understanding for the proper understanding of the specimen, and we can identify the correct name, and we know to do for the attribute of the organisms. And so that is a real understand nature. And after that, we can making for the conservation and the use of the nature. So this is one of the flow and just nature, but change to understand the nature. And the next is for the conservation. So it's a background. We need so many knowledge of the understanding for the history of the earth, or understanding of the natural environment, also local natures. And so this is very important for the museum activity. So, but not once. We repeat it again for the each 10 years or each 5 years. We checking for the look for the how to change for the environment. That is a very important point of view of the monitoring. Okay, next is I want to talk for the taxonomy of the beetles. So this is my beetle history, the beetles, and so I start to the undergraduate student. My supervisor Takehiko Nakane, so he gave me for the one of the subject is the taxonomy and the systematics of the history day, because for the 19th century, so it's a Englishman, it's George Lewis studied for the Japanese history day. After that, nobody studied the Japanese history day. And Professor Nakane gave me for the very nice subject. And after 40 years, almost 40 years, I have been for the study of the history of the beetles. Yeah, I usually use a bait trap. And so he is a Polish entomologist, is Dr. Mazur. He is the authority of the history of the world. And he visited for the Shiretoko in Hokkaido, and we collecting together. And this is for the collected by history beetle by bait trap. Some history beetle like such a costa is a strange surface. Also, this is Margarinotus saprinus is a history beetles collected by bait trap. It's a carcasses. So history beetle eat maggot is a larvae of the Diptera, and history also living in such habitat. This is a rubbish dump in Saharin. So under here is so many history there. Yeah, you can see this is a Diptera maggot, and this is a history there. So they picking up from here and eat. Yeah, so this way. So this is not one species. This is Margarinotus. This is Saprinus. So they eat together for the one maggot. So it's a dung or feces is attracted to the Diptera, and the Diptera lay the egg. And inside of the dung, so many maggot. So history they come to also is a dung. This is cow dung. So in the photo is from the Flores of Indonesia. It's a very big history there. So this is one of the tropical species. I think this species is distributed also in the Philippines. Yeah, this is the Japanese species, the Merohister and Atlas. Coastline of the beach area, also there's so many dead body of the seabird and the fish. And this is a literal Hysterics beetles, only for the distributed for the beach. Also, we can find for the underbug. Yeah, here there is so many larvae of the beetle or the fly, and you can collect these very depressed bodies and Hysterics beetles. Sometimes it's a cylindrical species. This is a predator of the Scolitidae, is a chamber beetles. Also, we can find Hysterics beetle in the nest of the ant. The beetle eat to the larvae of the ant. Yeah, very curious shape of the body. It's a very big Hysterics beetles, 8 milli. So we can find these near Sapporo mountains. It's Maruyama, 1997. Yeah, that is very surprised. So very, but not recently, but 20 years ago, we can find there is a very big Hysterics beetle inside of the ant nest. So this is a very small, just 1 milli, and the beetle living in for the soil. And usually we take for the litter soil and the shifting, and we collect such small Hysterics beetles, is micro Hysterics beetle. So the group is so many new species still in Japan, even in Japan also, it's Southeast Asia is so many new species. So is a look up the name, is we want to know for the name of the beetles. This is a taxonomy, is a special work of in the identification. It's a how to identification. Is the first is we checking for the picture book and literature, and is finally checking for the original distribution. That is very important. But sometimes it's already original description is very old and not so much description. On that case is we checking for the specimen reference collection, and is the last is examine the type specimen, and we are sure and confirm of the species name. And after with the taxonomist, we know for the knowledge and the result, and we publish the papers. So this is a Hysterics beetles, and so family characteristic is antenna. Antenna has an elbow-like, and 2 segments is the propygidium and the pygidium exposed. So this is we can separate from the other beetles. And about 4,000 species of the world, and Japan is about 120. Yeah, this is a picture book, and so we first checking for the what's the name. We want to look. We want to know for the name. Usually, we check for the book of the such picture book, but there's a very few description, and we checking for the as a professional taxonomist. Of course, we check for the other papers. On the paper is so many characters. Yeah, this is a dorsal side, and this is the ventral side. So we're checking about 80 for the characters and checking for the everywhere of the surface of the body. So this is a type of specimen and deposited in Paris, the Natural History Museum, Paris. So taxonomists visit for the European Museum, and we checking for, we want to sure for the name of the species. So this is my desk with binoculars, and this is original description. And for this is beetles, and we dissect it I dissected for the beetles because this is a male genitalia. It's a very important for identification. And so I take up pictures by digital camera and trace, and we make for the illustration of the genitalia. So this is very important for the paper of the taxonomic paper. So this is one of the examples, is a photo of the outside. Also, we use SEM, it's a scanning electronic microscope. And this is a result of the male genitalia. And sometimes for the female genitalia, this is a spermatheca. They keep a sperm inside of the body. Yeah, now is a number of the species about, in Japan is about 120. So this is all is subfamilies. So we can check for the numbers. So this is our distribution table. Sorry for the Japanese, but this is a local prefecture or district names. And this is a species name. So we making such a table, and we try again to collect for the new record for the beetles. Finally, we can make a map of the species. So this is the most common Hysterics beetles that is in Japan. Of course, I'm mainly focused on the Japanese fauna, but in Asia, not so much for the specialist of the history there. I'm also interested for the Russian side, it's Sakhalin and Kuril Island. And I visit for the Taiwan, and recently started the Philippines and Malaysia, Indonesia, and New Zealand. This is my interest area of the north to south. I visit frequently for the Asian area and the collecting for the history there. And I want to make a revision of the Asian history to beetles. Yeah, this is an example of the expedition of the Sakhalin I use for the bait trap and relate for the some friendship of the Russian people. So this is a graduate student. He is a special of the diphtheria. And this is the result of the bait trap. It's so many history to beetle and Staphylidae and Hydrophilidae. So this is the Indonesian photos. And so Dr. Mariamar, he set his flight intercept trap. This is a very effective tropical area. Just is a file case of the plastic one. So we making such small trap, and beetle fly and attacked seed and fall down its waters. But it's very effective. So this is a sorting, photo of the sorting. Yeah, this is a result of the phytotype. And this is a history there. And so I can make a specimen. Yeah, it's so many history there. This is a very good result in the tropical forest. Yeah, phytotype is very effective in the tropical area. So result of the Indonesian history there, it's Indonesian history is very diverse compared with the Japanese one. The very big, also very small one, and not so much worked. And still there is so many new species and sometimes a new genera also. Yeah, if I want to study for the Indonesian fauna of history there, so I have to visit two museums, the Amsterdam Museum. This is Europe. So this is the Indonesian Bogor Museum. Of course, it's a Indonesian collection. Indonesia is of course, but before World War II, there is Netherlands colonized in Indonesia. So many specimens in Europe. So it's mean for I have to visit the field and two museums, is Indonesia and it's Netherlands. So this is research of the exhibition field work, but also I visit several for the museums in Europe and the US. So study of the history, they still so limited for the adult, and the pupa, egg, larva, and pupa still unknown. So I hope to some next generation people is studied for the larvae of the history. There is so many new finding. So last is a collection. It's a very shortly I introduced. So I'm now working for the Hokkaido University Museum. And so this is my collection of the history day. So I keep to the history day such style. It's an insect box, and we said it's insect tray by papers and the different size. Yeah, our collection is a very old history. This is old building. About 120 years ago, it's a start to the Entomological Institute. So Professor Matsumura is a first professor. He's described so many orders, and he's one of the pioneers of Japanese entomology. And his discipline also are entomologists and taxonomists. So he's Dr. Matsumura, and Dr. Shiraki, he started to do Taiwan entomology. Yeah, it's an old photo. We keep it about 100 years for the, we keep the collection. Yeah, this is old collection. And some collection has a show for ecology of our life style of the some moss. And we keep to about 10,000 holotype in our entomological collection. Sorry, only Japanese. He's my professor is Takehiko Nakane, and his collection also we received, and about 1,000 boxes is only Beetle collection, but it's very nice collection. So this is our collection room, and there is a cabinet. Each cabinet has 48 boxes, and yeah, and our boxes has a cover of the wood. So because protect for the light, and open, and there is a insect tray and the inside of the specimens. And the insect tray is separate to the such size. It's much easier to move from the box to box because insect tray has a just unit, so more easier. So move to one by one of the pinned specimen. Insect tray has a species name, and P4 is meant for the Pararctic number 4 is a Manchuria element. And the Korea, Siberian element is Korea. So P4 is Japan. This is our style. Oh, it's not so good focus, but you can see this is a male genitalia, and so other old labels and identification label. So keeping the specimen is very important because if you conduct to the survey of the fauna, but after, so we have to check again for the identification of the species. That's case you need for the specimen for the confirmation. So the keep to the specimen is very important for the for such inventory work. So this is my homepage. Is Emamushi. Emamushi is a Japanese name of the history of Beatles. So this is a taxonomic. So you can click and you can find the subfamily level, and the tribe level, and the genus level, and click also is you can reach for the species level. So there is a specimen examined or material or sometimes description, and for the figures. So this is Atlas philippinensis, is a species described from the Philippines, it may be first from Philippines, but it's very old, 1868. So it's a sketch of the outside of the external characters, and the SEM photo, and male genitalia. So usually we use only for the line art in a paper, but on the website you can show, we can show for the it's a photo of the original of the male genitalia's photos. So this is very convenient. So taxonomy have a specimen and making study for the paper, but also so website is very convenient for taxonomy. So distribution map. Okay, the collection side is very short, but I introduce you for the sampling method and taxonomy and collection. So thank you for your attention.
Thank you, Dr. Ohara, for that very interesting talk. So you may now, to our participants, you may now start posting your questions for Dr. Ohara. So probably I'll start with asking Dr. Ohara. As I have noticed, most of your slides, of course, it's normal, especially for East Asian. I think it's quite normal for East Asians to have their native language or their local language in the way they present, of course. Not only because of nationalism, but I would like to ask Dr. Ara, how important is it to use your native language to educate people when it comes to biodiversity?
So maybe for the Philippines, so you are very common for the English talking and communication, but you know, Japanese is we have a terrible English and not so good. Is everybody speak for the broken English, and usually we use only for the Japanese language, and so each of species of the insect has a Japanese name. Yes, and also Latin name. But so we are very good arrangement for the Japanese name of the organisms, and we are only for the communicated to the for the Japanese language of the biodiversity or biology, something like that. So yeah, maybe it's important for the nationality or something keep to the very cultural keep of the thinking or some behaviors. I'm not a patriot, but it's anyway for the Japanese language. Yeah, we are very useful and keep to the such situation.
Thank you, Dr. Rara. Another question for me is, at least for my students, of course, to understand is, how important is identifying your species correctly to other studies that are connected. Of course, you're doing taxonomy work, but how important is good taxonomy to other studies such as ecological studies and other related fields of studies?
Okay, it's a very important, and the taxonomist identified name. So the species name will be the sure. So if for you know for the species name, correct species name, you have so many attributes because the species, when the species come to the outside for the other adult, and anyway for the you can check for the paper, use for the species name. So you got it to so many useful information. Sometimes a species will be a pest or sometimes will be a medicine. That is a more, how to say, good for the human of the like a more adaptive biology. That is one of the point of view. And but of course there is a useful organisms or useful very bad animals, but humans don't know real relationship of the organisms. And we are more need knowledge is a relationship of the insect and the plant or soil bacteria, something like that. Anyway, for the biologists to keep to the study, and sometimes we can find it's very nice for the network of the organisms, and sometimes useful, but sometimes not.
Thank you, Dr. R. Hey, there's a question here in the chat box from Arne Binti Sapaat. So do you face any pests attack in your collection? And if you do, what kind of treatments you apply on the damaged collections? Thank you.
Yes, so yeah, our collection is a very long history, and several times we attacked for the dermis beetles, and so unfortunately we still use for the naphthalene is chemicals. It's not so good for the human body, but so it's a 2.5 million specimen. We cannot control without for the chemical naphthalene. And we also use for the refrigerator, it's minus 20 degree and 3 weeks, and we killed to the pest beetles. Especially for the somebody donated for the collection, that is very, really careful, and each collection is killed for the pest under 20 degrees temperatures. Anyway, we keep to the, still we use for the chemical one.
Thank you, Dr. Ohara. Another question, so before that question, question from Eileen Vitug. So besides morphology, have you observed behavioral characters to determine taxonomic affinities in Coleoptera?
Yeah, and yeah, fieldwork is very important because we can observe for the behavior. And so some group of the Hysterics beetle, so their cooperative behavior is very strange. Usually male and female is cooperated with a mount, but some beetles is a bite of male bite female leg. That is the only special tribe. So if I observe such behavior of the courtship, so I can identify it easily to the tribe history. That is yeah, there is experience. Yes.
Thank you, Dr. Ara. Another question here from Fred, a science communicator. This is a citizen scientist, no? So have you observed subspeciation in hysteridids between different elevations in the same location? So same species but different subspecies. We found on top of the mountain versus mountain and versus lowlands.
Yeah, Hysterids beetle is a predator of the maggot. And that is a habitat is a very short time and limited. For example, dung or carcasses is very easy to disappear. So it's mean for the beetle depend on the such is a limited resource. So it's mean for they frequently move to the one place to another place, like a behavior with just domestic fly. So they are very good flying, and so very easy to move. So it's mean for the unfortunately for the normal Hysterids beetle, not so much differences of the elevation of something, and not so subspecies, subspeciation. But is recently genus Anapleus, we can find is from the soil, especially for the how to say, like a carabide is a more cape, like a small cape. That is we can find is digging of the soil and very rocky singles, and we found is Anapleus groups. That is a very, very diverse of the speciation probably, but not well studied. So that's that genus probably getting for the subspeciation of the altitude. Yeah, depending on their groups.
Thank you once again, Dr. Ara. Another question here from Jay Borsilio. So regarding flight intercept traps, so what are the factors? So this is for the method. So what are the factors that affect the catch rate of specimens using flight intercept traps?
Sorry, intercept trap for the question is
Regarding the use of flight intercept
yeah,
traps. So what are the factors that usually affect the
a
catch?
factor.
So
Yeah,
what influences the catch rate, I think, when using flight intercept traps?
yeah, flight intercept is just its word is intercept. So because a beetle and flying in the early morning or evening, so they flying very low, almost just 10 centimeters, 20 centimeters on the ground. And that is very difficult to the collecting for the human, but is a beetle flying and just attacked and whole. That is an intercept trap. So I recommend it for the many intercept trap setting for the forest. You can collect this nice result.
I think
Yeah, yeah. Reason is just intercept.
okay. So thank you, Dr. Ohara. So from JB Tibig from Ateneo de Manila University, in doing insect sampling, do you also take into account the microhabitats where the insects are found?
Yeah, it's a insect. Usually, insect living in the microhabitat, very very micro. And so in the field, we can detect to the very strange microhabitat, and we guess to the some related to the fungi or some special plant. That is a very important for the point of view for the entomologist. And that is sometimes we are committee member of the environmental evaluation. So entomologist point of view is very, very tiny and microhabitat, and but is sometimes very important. Yes.
Thank you, Dr. Ohara. From Shun Cairo, can we DNA barcode dry museum insect specimens?
I'm not, but so my colleague is used for the DNA, and the not so much DNA barcoding, but for so usually graduate student study to the external morphology, also DNA, and they reconstructed phylogeny by molecular levels. And yeah, barcoding part is got the sequence, it's much easier. Yes, and we can do. And I'm not related to the barcoding, but yes, it's our laboratory is a graduate student and other professor did such work.
Thank you once again, Dr. Ohara. This is for this question is related to maintaining collections. So this is from Leonardo Salak from Pampanga State Agricultural University. With regards to the maintenance and control of preventing rotting in your collections, so what do you do to prevent rotting in your collections? And usually, how many years will your collections last?
How many years?
Yes. How many years do your collections usually last? Probably his question, how many years will your collection last before rotting or fungi infection?
Yeah, it's an old collection, all over about 120s. Yeah, and we keep it in good condition, especially the Hokkaido is a northern area. It's a climate is very similar to European one, and not so much humidities. And even for the last 30 years ago, we have no air condition systems in our collection room, but we can keep to the good collection. In the Hokkaido is a very good place for the collection. And yeah, still we keep to the good condition. Just we afraid to the pest of the beetles.
Yes.
Yeah, yes.
As far as I understand, Dr. Ora, because the temperature in Hokkaido is less humid, so I think you're doing less maintenance with your collection. It's just that the pests are the only problem that I think that you are facing. of course, compared to the Philippine collections here, because we are a very humid country, so we need to use many other equipments and other chemicals to
Yes.
preserve specimens. But of course, in Hokkaido, it's quite cold. So yeah, hope if we have that sub-zero temperatures, probably it would be easier to keep the art. But though
Yeah, very, very good for collection, but not so good for the human.
I understand, Professor. I don't know if you can answer this question. So from Giselle Christine Paclibar of Lyceum Philippines University. So this is a question for an entomologist. So may I ask your opinion in using seed predators as a biological control agent for weed species? For instance, its effectiveness compared to leaf-eating insects.
Sorry, I cannot pick up the, yes.
So may I repeat that question? So she is asking for your opinion.
Yeah.
Using seed predators
Seed predators.
as a biological control agent
Yeah.
in species. So in comparison to leaf-eating insects.
So you, as a comparison of the seed eater or leaf eaters.
Seed predators versus seed-eating versus leaf-eating insects.
And so he asked you the which is more effect
for biocontrol.
Biocontrol. So it's a eater is a pest.
Yeah.
So yeah, usually for the leaf eaters is a pest. Leaf eater pest is controlled by the wasp, parasite wasp. But seed eaters, I'm not sure for the. Yeah, I'm sorry for I'm not familiar with the biological control. It's
Yeah.
Especially for the such plant insect. Yeah, sorry.
Problem, of course. No. From Sultan Kudarat, Sultan Kudarat State University, Clizel. So her question is, I'm currently a part-time laboratory aide at school and the research study of my subject teacher. So she studies about the insect fauna of Lake Colon and in Mount Parker at Mindanao. Do you have some tips on how to easily spread the legs of each Coleoptera and the best way of preserving Coleopterans?
Sorry, maybe I'm tired for that, so I cannot become again for that. It's a fauna.
It's my fault. No, so to make her questions short, so she wants to know if you have some tips on how to easily spread the legs of beetles and what is your suggestion on preserving beetles?
Sorry, which one?
Crizel and Arranque at around 11:14 a.m. I can check the chat.
This one. It's a little. Yes. Lake.
She's actually just asking for tips.
Spread the leg. I see. Spread the leg. I see. Mm-hmm. Yeah. So probably not so important spread the leg. Of course, it's better. But if not spread, so we can identify it. Yes. And I don't know for the easy way. Anyway, we use for the forceps and the pinned, and each specimen has a keep such way.
And probably, Dr. Ara, what is the best way of preserving the beetles?
That is a dry specimen is a best for the taxonomy. But if you want to take for the DNA, you are keep to the different way for the as a 90% alcohol something under or temperature control. Yeah, dry specimen is a pin and dry specimen is easy for checking for the taxonomy.
Thank you, Dr. Ara. Now this perhaps let's have this as a last question from Brenili Agaton of St. Joseph Academy of from Antique. So they say that beetles are the most successful animal life forms on Earth. So why do you think that they are the most successful animal life forms on Earth with your studies on beetles?
Hard body and hard mandibles and get to small size. Yeah, it's a beetles is a hard body is so strong. And this is one of the reasons of the diversity.
So thank you once again, Dr. Hara. So to close this lecture, I would like to ask you to, what would be your advice to our students who wish to pursue similar studies such as you on beetles and other insects?
Yeah, I'm taxonomist. I'm recommended. So if you're interested to the some insect or other organisms, so maybe you choose one group and keep to the study is long times. You understand it for the groups and easy to get specialist. So there is so many human and being, but there's some groups only you are specialist. Nobody know others. So it's been for the very important role of the science. So maybe Philippines is so diversity, so biodiversity. So Philippines need to some specialist. Maybe you can choose is one groups and you study to keep to the studies. That is you can success for as a biologist something. Yeah.
Thank you, Dr. Ohara. So before, once again, thank you, Dr. Ohara, for that wonderful lecture and for answering the questions of our participants.